This video is a continuation of our discussion of metabolism of lipids. Being more specific, we will look at the details of how lipase hydrolyzes the ester bond of triglycerides. During our introduction to the chapter of metabolism, we discuss what are the components or the players necessary for that to work. We discussed that carbohydrates, lipids, and proteins will be our fuels, that in catabolism we will need some molecules of ATP for reactions that are endergonic to become estergonic. Then we also discuss how we need to use enzymes, and if we have enzymes, we also need to use vitamins and cofactors. We need to use electron carriers and carbon carriers. The objective is to cleave the ester bond of a triglyceride to produce a fatty acid that, upon activation, can produce molecules of acetyl coenzyme A. Those molecules will enter the citric acid cycle to release carbon dioxide and 8 electrons. The 8 electrons bound in the reduced coenzymes will enter the electron transport chain to pump protons from the matrix to the intermembrane space. When the protons return back to the matrix, that will help ATP synthase to release useful ATP molecules. Pancreatic lipase catalyzes the hydrolysis of triacylglycerols at carbon number 1 and carbon number 3. The active site of lipase contains a catalytic triad that is very similar to that one found in serine proteases. We can say that the peptide hydrolysis in a protein, it is mechanistically similar to that of the ester hydrolysis of a lipid. This is a good example for acid-base catalysis and also for covalent catalysis, where we're going to form a temporary covalent bond between the active site of the enzyme and the substrate. Because serine OH is not a good nucleophile, it will need to be activated by the protonation from the neighbor histidine amino acid. The alkoxide form is much better nucleophile. The aspartate residue helps to orient the histidine amino acid to be much better proton acceptor and also stabilizes the system by hydrogen bonds and also by electrostatic effects. Upon binding the substrate, the reaction begins with the nucleophilic attack of the alkoxide to the carbonyl of carbon number 1 or carbon number 3 to form an unstable intermediate tetrahedral that bears a formal negative charge on the oxygen that came from the carbonyl group. The generated negative charge is stabilized by some groups in the protein. When the tetrahedral collapses, it's going to form an ester, that is using the acyl group of the fatty acid and the oxygen of the serine. In addition, upon deprotonation of histidine, a 1,2-diacylglycerol unit is released. Here we can see the unit of 1,2-diacylglycerol and the proton that was taken from histidine and the fatty acid that is forming a temporary covalent bond with the active site of the enzyme. In order to do the hydrolysis of the ester bond between the fatty acid and the serine amino acid, now we need to use a molecule of water. Histidine now will take a proton away from the molecule of water, making hydroxide much better nucleophile. The resulting OH ion attacks the carbonyl carbon on the acyl group, forming another intermediate tetrahedral. Here we can see histidine is protonated by the molecule of water. Also, we know that that plus charge is stabilized by the neighboring negative charge of the aspartate amino acid, and the OH is much better nucleophile that attacks the carbonyl of the ester in the temporary covalent bond between the fatty acid and the active site of the enzyme. And this cartoon is to represent the oxyanion hole. The efficiency of this enzyme is due to the catalytic triad, the opportunity of the formation of a temporary covalent bond, and to the presence of those amino acids that can stabilize the intermediate tetrahedral in the oxyanion hole. The second tetrahedral collapses, releasing a free carboxylic acid, and the active site of this enzyme gets restored when histidine donates a proton to the serine lone pair of electrons. The fatty acid released will need to be activated by use of a molecule of ATP and coenzyme A, and that will be formation of a thioester bond. But I will have another video to explain that. And we can see that the active site of this enzyme is restored and can be used for a second hydrolysis of 1,2-diazoylglycerol or another unit of a triglyceride.